All right, we're on to our next section now, which is the contraction of muscles. So we're going to take a look again specifically at how skeletal muscles are going to contract, although the difference between skeletal and cardiac at least is relatively minor. And as I mentioned in the previous section, the overall mechanism or description of how these muscles are going to contract is known as the sliding filament mechanism of muscle contraction. So what's going to happen here is that the thick and thin filaments we described back in the previous section, we talked about thick and thin filaments that make up the myofibrils of the muscle cell itself, are going to sort of grab onto and walk past one another. And as they do that, that's going to cause an overall shortening of the muscle fibers, which collectively then leads to an overall contraction of the muscle itself. So again, we're dealing with actin filaments, we are dealing with myosin thick filaments, we are dealing with troponin and tropomyosin, which are the we call accessory proteins on the thin filament down here as well. Really four main proteins that we're going to be dealing with in this section on muscle contraction. The actin filaments themselves, these guys here, kind of looks like a twisted string of pearls, something like that. Each one of these has an active site on it. Myosin binding site maybe is a better way to say it. A myosin binding site on each one of these actin proteins here. And in this picture you can't see them because they're covered up. And so what's happening here is that the troponin is covering up the active site on the actin filament, the myosin binding site on the actin filament. And tropomyosin is also going to be involved with helping to remove itself from the myosin binding site on the actin protein. So there's a kind of a cascade of effects that's going to occur. Once a muscle is told that it needs to contract, there is going to be kind of a sequential pattern of events that occur that eventually lead to the muscle contracting. So the accessory proteins are kind of like the sheath, if you will, that prevents the binding of myosin to the actin proteins until contraction is ready or is necessary. So once contraction is going to begin, what's going to occur here is that the accessory proteins, and again, namely those are the troponin and tropomyosin, they are going to be pulled aside, and they are going to expose the myosin binding site on the actin proteins to the myosin head itself. So in this picture, now we have these binding sites, myosin binding sites on the actin proteins, which can be grabbed onto by this myosin head, but only if the tropomyosin and troponin proteins are pulled aside as well. So again, in order for myosin to bind the myosin binding site on the actin protein, there's going to have to be a grabbing and pulling of this troponin protein, which is then going to, as a result, pull on the tropomyosin, kind of the blue line in this picture. And once that occurs, effectively what happens is now we see these actin myosin binding sites on these proteins here. And so once they're there, the myosin heads can bind to the active sites. They can bend. So as we see, we have a binding and then a bending or a flexing of the myosin head. This is, again, the bottom part of the hockey stick, if you will, is going to flex this direction. And as that occurs, it's going to cause an overall contraction in the left to right direction on this picture. The final thing that's going to happen here in this picture is that in order for the myosin head to release from the current myosin binding site on the actin protein, there's going to have to be an expenditure of ATP. And we see ATP being spent right here to allow the myosin head to release, straighten out, and then bend forward and grab onto the next myosin binding site on the next actin protein. So that's where ATP comes in. Now, muscle contraction is a very, very energetically expensive thing to do. The place where glucose needs to be spent in order to make ATP to cause muscle contraction is going to involve the releasing and flexing of the myosin heads to bend forward and grab onto the next myosin binding site on the next actin protein, like we saw in the previous slide. So what occurs if this is happening in both directions, because what we saw over here was only contraction in the left to right direction, but just as easily there could be contraction in the other direction as well. What's going to happen here is these alternating bands of thin and thick and thin and thick and thin filaments are going to walk in both directions, and so there's going to be a shortening this direction and this direction. So the overall sarcomere, the overall functional unit of contraction itself, the entire thing, is going to shorten. And if the entire thing shortens, if the entire muscle shortens along its entire length, then you're going to have movement. Right? The contraction of the muscle is going to lead to the either flexation or extension of perhaps an arm or a leg, biceps, triceps, quadriceps, hamstring, flexing, relaxing, extending, all those kinds of things happening together. So we're going to kind of back up now. What we just saw was the overall pattern of muscle contraction. You really want to make sure if if I have one bit of advice for this chapter, and chapters like this one, is that if you want to learn the mechanism, the pattern, the pathway, the process of muscle contraction, you want to write it out. You want to be able to draw a flow chart. You want to be able to explain this to somebody else. You want to talk about how the troponin, triple myosin, and actin is all related to this process of skeletal muscle contraction. What we're going to do now is we're going to back up one step. We're going to make the story a bit longer to see kind of like the prequel part of the story, if you will. So in order for skeletal muscle to contract, of course, there has to be some sort of control or message sent from the nervous system telling the muscle when and how much it needs to contract. Now, we are dealing here with skeletal muscle, so for the most part, this is going to be voluntary activity with the possible exception of reflexes, for example. There are going to be nerves as part of the peripheral nervous system, motor neurons, that are involved with synapses, which on the other side of the synapse, there's going to be a skeletal muscle. And so wherever you have a neuron joining up with a muscle, we have what's called a neuromuscular junction. So we have this joining of neuron and muscle across a very small synapse, and that's where the communication is going to occur. So what's going to happen here in a second is that we're going to see a neuron which is fired. It's going to eventually stimulate a muscle cell to contract. What we want to see is what happens between the neuron firing and the contraction of that skeletal muscle. The sequence of events that occurs up to, we talked about before, with the various proteins in the muscle fibers and the shortening, sarcomeres, and so forth. So in the case of vertebrates, like we've talked about before, in our case, in the case of humans, in the case of mammals and birds and those sorts of things, there is going to be a chemical communication from the neuron across the synapse to the muscle cell waiting on the other side. And the chemical communication we're dealing with here is a neurotransmitter, like acetylcholine, like we've seen before. So in these pictures here now, we have the end of an axon joining up with 1, 2, 3 different muscle fibers. And again, muscle fiber is synonymous with the term muscle cell. So this one neuron controls 1, 2, 3 muscle cells. But if we zoom in on one of the tips of one of these axons, what we see here is a synapse. We see the neuron. We see the postsynaptic membrane of the muscle cell. And what we're going to take a particularly close look at is what's going on inside that neuron. So when this neuron is excited, when it reaches its action potential, or when it fires, another way of expressing the idea of action potential is by saying a neuron fires, there is going to be a release of neurotransmitters from these synaptic vesicles. Remember vesicles back from chapter 3 when we talked about parts of cells. These vesicles contain neurotransmitters like acetylcholine that once they are released into the synapse is going to cause an excitation of the muscle cell. And that's going to kind of trigger or kick off the next in the series of events that leads up to the muscle cell contracting. Now, in this case, neuromuscular junctions are always excitatory. There is no inhibitory postsynaptic potential from neuron to muscle cell. Whereas with a neuron-to-neuron connection, you can have both EPSPs, excitatory postsynaptic potentials, and IPSPs, which are inhibitory postsynaptic potentials. In the case of neurons to muscles, they're always excitatory. Okay, so what's going to happen here now? This is the prequel to the idea of muscle contraction that we saw before. All the way up here in the neuron, we have a nerve impulse or a firing traveling down the axon to the point where it's going to cause the release of acetylcholine from these vesicles into the synapse. Now, once this acetylcholine goes into the synapse, it's going to bind to receptors in the plasma membrane of the muscle cell, and that binding of acetylcholine to those acetylcholine receptors is going to cause a release of calcium ions from the sarcoplasmic reticulum. We talked about the SR is the place where calcium ions are stored. They don't get released until there is neurotransmitter in the synapse up here. Once we have that neurotransmitter, now we can release the calcium ions from the SR. We can pass those calcium ions in, around, among, and through the muscle cells, so they get all throughout the muscle cells via the T-tubules. All right, so the effect of the calcium ions. Right? So these calcium ions in this picture that are being released in and among those muscle cells, what these calcium ions are going to do is they are going to bind to troponin. So if we're going to go way back here for a second, I'm going to go back to this picture right here. Here we've got a relaxed muscle, a single tiny section of a sarcomere from a piece of relaxed muscle. But what you want to imagine here is all of a sudden calcium ions being thrown at this picture. There's calcium ions everywhere. And what those calcium ions do is they're going to bind to troponin. Calcium ion fits here, and here, and here, and here. And once those calcium ions fit into that troponin, troponin is going to be pulled aside. And since troponin is also attached to tropomyosin, once you pull on troponin, tropomyosin is along for the ride, and you're going to expose those myosin binding sites on the actin proteins. So the troponin and tropomyosin have been removed here because of the activity of those calcium ions. That's kind of the beginning part of the story that we didn't see before when we talked about muscle contraction. If we want to look at a bigger scale as it pertains to muscle contraction, we can also take a look at what are called motor units. A single motor unit is a single motor neuron. Again, this is part of the peripheral nervous system, somatic nervous system in this case, because we're dealing with skeletal muscle. It's a motor neuron and all the muscle fibers or muscle cells that it controls, or that it innervates, the ones that it controls, right? So whenever that neuron fires, whenever that neuron reaches its action potential, all of those muscle fibers are going to contract. So it's an all or none sort of a thing. If the neuron fires, then all of its muscle cells are going to contract using the same mechanism we just talked about, right? So we release neurotransmitters, that releases calcium ions through the T-tubules. Calcium ions cause troponin to move aside, which causes tropomyosin to pull aside, which allows myosin heads to bind to active sites on the actin protein, okay? So that's for a single motor unit. Now, if you look at muscles that have very, very precise control, so I'll give you an example here. The muscles that move your eyes. Think about the degree of precision that it takes to look and fixate in on a single target. That's extremely, extremely precise control over some very, very tiny muscles. Those are some very, very small motor units. You've got almost one neuron per muscle cell. It's not that extreme, but it's pretty close. One neuron, one muscle cell, very, very great degree of control over those muscles. On the other hand, if you look at muscles that maybe need more power but less precise control, then you could have larger motor units. You could have a single neuron and maybe 100 muscle cells that it controls, because the precision doesn't need to be there. If you're looking at the muscles that control your thigh, the thigh muscles, if you just look at your thigh muscles, those are relatively large motor units because they're big muscles, they're very powerful. They don't need a lot of real precise control. You can get away with having fewer neurons per muscle cell, and so that would be a larger motor unit. Now, recruitment is what occurs when you need to start adding more and more power. So if you go to pick up a pencil that's sitting on the table, you don't need to use every single motor unit in your arm to do so, because that pencil is not that heavy. And if you did use every single muscle to pick up that pencil, you'd look really silly doing it, because the pencil would go flying up into the air and you'd look like you're having some sort of a spasm. You only need to use enough power to do what you need to do. So when you go to pick up the pencil, you're going to use just enough motor units in the muscles in your hand, in your arm, and your shoulder to pick up that pencil, no more, no less, just enough. So if you go to pick up, instead of a pencil, if you go to pick up a 50-pound weight, now clearly the 50-pound weight is going to weigh a lot more. You're going to need more power to pick that up. So now you're going to recruit more motor units to the table, and that will hopefully allow you to pick up that heavy weight. So instead of just using 3 or 4 motor units, now we're going to use 300 or 400 motor units to generate a lot more power and hopefully allow you to pick up what you need to pick up. So again, this is an idea of a case where you've got relatively few motor units activated. Tapping your toe does not require a lot of power. You can very easily get by with using just a few motor units and be perfectly fine. If, on the other hand, you're going to run, running you're going to have to generate substantially more power, so now you're going to want to use more motor units. Instead of just using this one motor neuron and the few muscle cells that it hooks up to, now you're going to use all 4 of these motor neurons and all of these muscle cells to generate power. So again, we've got here in this picture a neuromuscular junction. This is a motor neuron coming in with its axons, joining up with individual skeletal muscle fibers. Kind of a cool picture. Again, within these skeletal muscle fibers, we could zoom in even further and see things like the myofibrils, the thick filaments, the thin filaments, sarcomeres, and so forth. Now, when it comes time for muscle contraction, there are 2 other things in addition to the muscles themselves that we've talked about that are very important. There needs to be calcium ions, which are the ones that allow for troponin and tropomyosin to expose the myosin binding site on the actin protein, and also ATP, because the ATP is required for the myosin heads to let go or release from the myosin binding site on the actin once the need for contraction is over. So that's kind of the minimum requirements you need to have. The action potential as it moves down the muscle fiber into the muscle cell is again going to cause those calcium channels to open up. Those are in the SR. The calcium ions then flow into the surrounding thick and thin filaments, allowing the troponin and tropomyosin to move aside, and we've kind of talked about that story a couple of times. But that's where calcium comes in. Calcium binds to troponin, tropomyosin moves away, myosin can then bind to actin, and you start seeing that shortening that we talked about previously. Of course, you also have to have the ATP for that myosin protein to bend forward and grab onto that next actin protein as well. Let's see. All right, so as long as we have the exposure of binding sites, there is going to be contraction taking place. As long as you have ATP, there's going to be contraction taking place. So as long as we have these things there, the myosin heads are going to continue to walk their way along those actin proteins. They're going to bend and release and reattach and bend and release and reattach and bend and release and reattach. And that's going to continue to contract this muscle fiber. The myosin head itself, the part of the hockey stick that bends, that also is very, very important because that's where we have the myosin actually releasing from the actin protein to allow it to reach forward and grab onto the next actin protein. All right, so when it's time for the muscle to relax, we don't want to contract the muscle hard all the time because that's very uncomfortable, can be very difficult to move if you have like a cramp, for example, where the contraction is not permanent, but it's more long-term than you really want it to be, and it becomes painful and so on. The calcium ions have to be taken back up into the SR. The calcium ions come off of troponin, and once calcium is removed from troponin, then we see tropomyosin going back over the myosin binding sites on the actins, and all those accessory proteins go back to their original location. Right? The accessory proteins are back in place, the myosin binding site is now covered up, and we're back to a relaxed state. Now, a situation called rigor mortis. You may have heard of rigor mortis before. This is a case where muscle contraction becomes permanent because there's no more ATP. And so we talk about cross bridges. The cross bridge is what happens when you have the link between the myosin head and the actin protein. If there is no ATP, that link between the myosin head and actin protein can no longer be broken, and that muscle is going to stay contracted in that state until there is ATP there or until the muscle begins to decompose, as we'll see. So when someone dies, within 3 to 4 hours or so, there's going to be a stiffness. This is the reason why dead people are called stiffs, I suppose. After a couple of hours, the muscles are going to kind of lock up in the position they were in when that person passed away. And since they are no longer manufacturing ATP, think about where ATP comes from. It comes from cells that are doing aerobic cellular respiration. If there's no ATP being generated, then we can't put the accessory proteins back. We can't break the cross bridges between the myosin and actin. And the muscle is going to stay locked up where it was before. After 2 or 3 days beyond that point, when the rigor mortis has first begun, there is going to be a relaxation again because the muscles are going to start to break down. Once the muscle protein breaks down, then everything releases and there's no more contraction of those muscles. Let's see. All right, one more slide in this part of the lecture, then we'll kind of wrap it up. The other kind of situation that's not abnormal but can be somewhat detrimental is if you have too many calcium ions entering into a muscle cell. If for whatever reason, if the SR has some reason to release too many calcium ions into a muscle cell, the proteins can actually be destroyed. The proteins of the muscle cell can actually be destroyed. And if the muscle cells are killed, if they're dead, then there are fewer cells available for contraction, so the muscles actually get weaker. Dystrophic means they're breaking down and no longer functional. Therefore, the muscles are getting weaker. There are several different types of muscular dystrophy. One of the more common ones is called Duchenne muscular dystrophy, where we have a defective gene for a protein called dystrophin. And dystrophin sort of works like a control over the amount of calcium that actually goes into the muscle cell when it's time for the muscle cells to contract. So if for whatever reason, if you inherit a faulty copy of the dystrophin gene from one or both of your parents, then when your muscles contract, every time your muscles contract, you're dousing them with calcium ions that are in high doses detrimental to them, and so those muscle cells begin to die off and they weaken, and eventually it becomes extremely debilitating for individuals that have this type of muscular dystrophy. But again, it goes back to the idea of proteins coming from DNA, and if you don't have the right DNA, if you don't have the right genes or the right stretches of DNA, then it's very difficult to make the proteins that you need and for them to function the way they need to function. All right, so we're going to cut off here, and we will come back with the third section in the lecture on the muscular system.